Welcome. In the previous session, we completed objective on body fluids and circulation. And in this session, I'm going to begin with a new chapter, excretory products and their elimination. Now, animals during metabolic activities, they produce a lot of materials that will be of no use in the body and they have to be eliminated. So during these metabolic activities, or maybe because of ingesting those materials in excess quantities, the animal will accumulate ammonia, urea, uric acids, okay, and even carbon dioxide, or maybe water, right, and even ions like potassium, sodium, okay, chlorides, phosphates, sulfates will be present, right, and these products which are present in excess quantities or that other toxic substances has to be eliminated from the body. Now, different organisms have developed different organs to eliminate these excess or waste materials or toxic materials from their body. Now, let us see what are all those excretory products and how the animals developed different organs to eliminate these waste materials, and then later we will see how the excretion takes place in human beings. So now let's see. So here there are three major nitrogenous wastes that are produced. So three major nitrogenous wastes. So they are one, ammonia, then urea, and then uric acid. The animals who produce ammonia are called aminotelic. So animals who produce, or I'll write who excrete ammonia, so they are called aminotelic animals. And the animals who excrete urea, they are called ureotelic animals. And the uric acid, so animals who excrete uric acid, they are called uricotelic animals. So they are called uricotelic animals. Right. Now we will see some examples, okay, for the aminotelic animals. Examples for aminotelic animals. So these include many bony fishes. Then they also include aquatic amphibians, okay, and then aquatic insects, etc. Right. Now coming to these ureotelic animals. So these ureotelic animals will include terrestrial amphibians, okay, then marine fishes and mammals. So I'll mention here. So examples for ureotelic animals. So these include one, marine fishes like sharks and all. They produce urea, and some of them even store urea in the body to maintain osmolarity. And the next one, it also includes terrestrial amphibians, and then mammals are the one who release the nitrogenous wastes in the form of, okay, urea. Then coming to the uricotelic, so examples for uricotelic animals. So these include insects, then also includes land snails. Oh, there are others, okay, aquatic also. So in case of land snails, they produce okay, uric acid. Then the reptiles and birds. So all these will be releasing the nitrogenous wastes in the form of uric acid. Now, if we see right these three nitrogenous wastes, okay, here out of the three, the ammonia is highly toxic when compared with the urea and uric acid. Okay, so I'll mention the order of toxicity. Order of, okay, toxicity. So ammonia is highly toxic, and this is okay much toxic when compared with the urea, and urea is toxic when compared with uric acid. So I say out of these three nitrogenous wastes, the least toxic nitrogenous waste is uric acid. So this is least toxic. So this is the order, okay, of toxicity. And here for removal of ammonia, a lot of water is required, as most of the aquatic animals don't have a problem of water. They eliminate the ammonia directly as ammonia, right? Whereas in the case of terrestrial animals, what happens, even this ammonia is formed, okay, so this is the basic product, and that will be converted either into urea or uric acid. So in terrestrial animals, the ammonia will be released in the form of urea or in the form of uric acid. So for this urea and uric acid, less amount of water is required. So in the case of uric acid, it will be the very much less water is required to remove this uric acid. Okay, and highly water is required for this removal of ammonia. Okay, so so we have come to a clear that ammonia is highly toxic, and uric acid is least toxic. Right. So here, if I compare this the urea with the ammonia, so how much it is toxic? So this urea is 1 lakh times less toxic than ammonia. So it is 1 lakh times less toxic than ammonia, right? So this urea, so I told you that most of the aquatic animals, they release, okay, the produced ammonia directly, okay, by using the water, whereas terrestrial animals will convert this ammonia into urea and uric acid, because in case of terrestrial animals, the availability of water is less when compared in case of aquatic organisms. So it is not like always the aquatic organisms, okay, right, will eliminate the nitrogenous wastes in the form of ammonia. In some cases, they even convert, okay, or convert this ammonia into urea and excrete the nitrogenous wastes in the form of urea, like in case of shark fishes. Okay, so now let us see this urea formation. It takes place, okay, in land animals, especially in an organ called liver. So there is a cycle, okay, in which the ammonia will be converted to urea. Okay, so ammonia is converted to, okay, urea. So where you can take right. So the ammonia is converted to urea in the liver, and this conversion has a name. So it is a cycle, and we call it Krebs ornithine cycle, and this takes place in Krebs ornithine cycle. Okay. Krebs ornithine cycle. The Krebs ornithine cycle, you can also simply call it a urea cycle, or even you can call it right, Krebs Hanslite cycle. So I'll just briefly discuss about this Krebs ornithine cycle, how it takes place in the right, the liver cells. So that's Krebs ornithine cycle. You can also simply call it a urea cycle, or you can also call it a Krebs Hanslite cycle. The ammonia which is formed inside the cells will, okay, react with this H plus, and it results in the formation of ammonium ions, and these ammonium ions, okay, will travel to the liver in the form of, so this will be, okay, transferred to liver in the form of amino acids like alanine and glutamine. In the form of alanine and glutamine, and once they reach, okay, the liver cells, okay, the ammonia will be released, I'm sorry, the ammonium will be released, and this will enter into the mitochondria of the liver cells. So here in the liver cells, it is the mitochondria involved in, okay, urea cycle. So the mitochondrium will be involved in the urea cycle, right? So already in the mitochondria, there will be a substance that we call an amino acid called ornithine. So I'll talk about it later. So now this, okay, released ammonium will enter into the mitochondrium, and this will combine with carbon dioxide. So I'm giving a brief, okay, idea about the urea cycle, and this will be catalyzed by an enzyme called carbamyl phosphate synthetase. So in short, I'll write CP synthetase. I'll just mention the full form here. The CP synthetase stands for carbamyl phosphate synthetase. So carbamyl phosphate synthetase is the name of the enzyme. So this will react, okay, in this presence of the enzyme, and it results in the formation of okay, carbamyl phosphate. So it results in the formation of carbamyl phosphate. So during this reaction, there will be, okay, two ATP molecules will be utilized, which will become 2 ADP plus PI. So this reaction takes place inside the cytoplasm of the mitochondria. Okay, and when this carbamyl phosphate is released, as I told already, the mitochondria has one more, okay, substance. It's called ornithine. So the ornithine and this carbamyl phosphate will react with one another in the presence of ornithine carbamyl, right? So in the ornithine carbamyl transferase, I hope it's clear. Transferase is the enzyme, right? So this ornithine will react with this carbamyl phosphate, and in the presence of the enzyme ornithine carbamyl transferase. Right. So once this is completed, it results in the formation of another substance called citrulline. So this whole process takes place in mitochondria. Let's say mitochondrium. Okay, so it's taking place in the mitochondrium. So this citrulline, which is formed in the mitochondria, it will be right passed into the cytoplasm of the cell. So citrulline comes out of the mitochondria. Right now here with the aspartic acid. So in this case, the aspartic acid is involved, and this results in the formation of arginosuccinic acid. Formation of arginosuccinic acid, and during this right, it will use one more ATP molecule. So ATP molecule is used, which results in the formation of arginosuccinic acid. And the enzyme involved here is arginosuccinic synthetase. I'm not mentioning okay here, or anything I'll do here, I'll mention. It's a arginosuccinic synthetase. Arginosuccinic synthetase is the enzyme involved in this, okay, citrulline and aspartic acid to form arginosuccinic acid. So this arginosuccinic acid as there is no space, I have written again, okay, here. Right, so the reaction between this, okay, ammonium and carbon dioxide in the presence of CP synthetase results in the formation of carbamyl phosphate, and right, this carbamyl phosphate will react with the ornithine in the presence of ornithyl carbamyl transferase enzyme, and results in the formation of citrulline. So the citrulline, okay, right, along with the aspartic acid, it results in the formation of arginosuccinic acid, and here also there is one molecule of ATP is utilized in the presence of enzyme arginosuccinic synthetase, and this results in the formation of arginine, and also it releases, okay, during this process the fumaric acid. So it releases the fumaric acid. Now this arginine will be converted to, right, back to ornithine in the presence of an enzyme called arginase, and during this it releases, okay, urea. So it releases, okay, urea, and finally it gets converted to form the ornithine, and the ornithine again reacts with another carbamyl phosphate, and the cycle continuously goes on. So this is just a brief idea about the Krebs ornithine cycle. So in this cycle, important things that we need to keep in mind is that there are actually two molecules of, okay, ammonia is involved, and it has resulted also in the usage of three ATPs and one molecule of carbon dioxide. So plus one carbon dioxide molecule plus three ATPs are utilized. So here this is the thing which we have to keep in mind. So how many molecules of okay, the ammonia utilized here, two molecules, then one carbon dioxide and three ATPs are involved in this reaction. So this is about the Krebs ornithine cycle briefly. Next we'll see about excretory organs in different animal groups. Right, so excretory organs in different animal groups. You see invertebrates, in invertebrates these excretory organs are simple tubular forms. So there they are just simple tubular forms. But coming to the vertebrates, so these simple tubular forms, okay, became complex, okay, and we call them complex tubular forms. So the vertebrates have complex tubular forms, right, and they together make a organ called kidney. So the vertebrates mostly have these kidneys for the process of excretion. So now let's look at different excretory organs in different animal groups. One, the protonephridia, and another one, nephridia, and then third one, Malpighian tubules, and the fourth one, the antennal glands or green glands. Antennal glands and green glands, and we have one more, the kidneys, that we will see, okay, later. Now flame cells, which are also called protonephridia, and these flame cells are seen in case of uh, the platyhelminthes, which includes, okay, planarians, and also in rotifers. So I'll say the flame cells example in planaria, in rotifers, and there are some annelids also, and also, okay, in amphioxus, which is a cephalocordate, okay, that so these are, okay, different groups in which we see the protonephridia. And okay, coming to this nephridia, we see in case of right, earthworms, and other annelids. Whereas coming to these Malpighian tubules, the Malpighian tubules we will see mostly in insects like cockroaches. And in case of antennal glands or green glands, we will see in case of crustaceans. So insects and crustaceans both they belong to phylum Arthropoda, and the crustaceans like prawns have this antennal or green glands. Okay, so we'll see the structures when we come to this protonephridia or the flame cell. So the flame cell, we'll see in the diagrammatic. So here I give just okay a simple diagram of a flame cell. So here these are all the protoplasmic projections, and here the cilia. So the movement of this cilia gives a appearance of a flame. So that is the reason why it has the name the flame cell. Here is the nucleus. So these are all cytoplasmic projections. This is a nucleus and this is a ciliary tuft. Okay, and this one finally opens into another duct. So this is how a flame cell looks like. Then coming to this nephridia, the nephridia have okay a nephrostome. So it will be showing further coiling. So there will be a lot of coiling here. I'm just okay showing a simpler one. So there are cilia here. So this is the nephrostome, the opening of nephridia, and this all is, I'll simply call it body of nephridium, and this opening is called nephridiopore. This is what the nephridium, singular, nephridia plural. So nephridium, how it is seen in case of earthworm, and the flame cells how you see in case of platyhelminthes. Okay, right. Primary function of this protonephridia is to maintain ionic and water balance, that is fluid volume. So what I call it osmoregulation. So I say that not only the protonephridia, so any excretory organ, the actually the primary function is to okay maintain osmoregulation, that is ionic and the fluid volume of the body. So that's all about excretory organs in different animal groups. Now we will see the excretory system in case of human beings. Excretory system in humans. Right. So the excretory system in humans, it consists of kidneys, so these will be one pair, and also has a ureters which originate from the kidneys, and these will be also one pair. And then it has a urinary bladder, and there is only one urinary bladder, and then urethra, that is also one. So in fact the urethra originates from the urinary bladder. Now these ureters, urinary bladder and urethra together they constitute a urinary tract. So all these together called urinary tract. Now the kidneys, coming to, we'll first see about these kidneys, okay, right, and then the ureters, urinary bladder and urethra. Of course here a simple diagram of that, the ureters, okay, so which open into the urinary bladder, and the urinary bladder continues as, okay, urethra here, right. So you can take, so this is the left side and this is the right side. So the left side kidney is slightly above when compared with the right side, or the right side kidney is lower than the left one, and it is because of a presence of liver on the right side. So here are the ureters, okay, the left and right kidneys, and this is the urinary bladder, and this is urethra. The dorsal aorta will divide and gives off branches called renal arteries to the kidneys. So the dorsal aorta divides and gives off branches to the kidneys, okay, so that is okay, so this is a renal artery. And at the same time veins also emerge out from the kidneys. We call them renal veins, right, and they open into the major veins that will be collected, okay, back to the heart. So here the renal artery, you will also find renal veins also. Here I'm not mentioning it, right. Now we will see especially about the structure of the kidney. So kidneys are mesodermal in origin, means they develop from the mesoderm germ layer during the development. And here these as I already told, the right side, okay, kidney is slightly inferior to the left side, okay, and then so the shape of the kidney is bean shaped. The shape of the kidney is bean shaped, and then color is reddish brown. The color is reddish brown, and size, coming to the size, we will see the size in case of adult human beings, okay, in adult humans. So what will be the measurements of the kidney? So it will be 10 to 12 centimeters in length, and then okay, 5 to 7 centimeters in width, and then 2 to 3 centimeters in thickness, in thickness, okay. So that's what, from here to here it is the length, that is 10 to 12 centimeters, and here to here it is the width, that is 5 to, okay, 7 centimeters, and the thickness will be 2 to 3 centimeters. So this is the thickness. So these are the measurements of kidney in the adult human beings. Then coming to the location, so where the kidney is present. So the location, we'll see the kidney between the levels of the last thoracic. It is present between last thoracic, okay, that will be last thoracic vertebra. Okay, so the last thoracic vertebra, which we take it as a T12. So T12, so T stands for that thoracic region. So between the last, okay, vertebra, that is a T12, and extends up to this third lumbar region, to third lumbar vertebra. Okay, so that will be L3. So I simply say that it will be from, and write L3, it will be from T12 to L3 is the location, and it is present very close to the dorsal side, dorsal inner wall. So in the dorsal side, so dorsal side is our back side, okay, and vertebral column is also present on the dorsal side, and it is present very close to the vertebral column, okay. So close to the dorsal inner wall of the abdominal cavity. So that's about the location here. And when we see, so this is the, say suppose the dorsal side and the ventral side, okay. Now as I said, the kidneys are very much close to the dorsal wall here, they are present, and extending between T12 to L3 vertebra region. Now here the dorsal surface of the kidney is attached to inner wall of the dorsal side, and the ventral surface of the kidney is covered by the peritoneum layer. So you'll find a peritoneum layer. So here is the kidney. So I said that the dorsal side of the kidney is very much close to the right, the inner wall of the dorsal side, and that is not covered by the peritoneum. Only on the ventral side of the kidney is covered by the peritoneum, is covered by peritoneum. Okay, so because of this, okay, the kidneys we call here as retroperitoneal kidney. So it's called retroperitoneal kidney, as the peritoneum covers only on the ventral side. So you can also use one more word for this, is extraperitoneal kidney or extraperitoneal kidney, right? And the weight of the kidney will be around 120 to 170 grams. Weight is 120 to 170 grams. And the kidneys are regarded as, so kidneys are homeostatic organs, as they maintain, okay, and they help in maintaining the homeostasis, so everything balanced in the body. And these kidneys are connected to, okay, sympathetic nervous system. So kidneys are connected to sympathetic nervous system, okay, right. So now with this I'll end here. In the next session we will see the longitudinal section of the human kidney and the complete anatomy of the kidney we will see in the next session.